Okay, good evening everybody, and thank you for coming for tonight's lecture. My name is Silvio Rizzoli from the University Medicine Göttingen, and I'm going to introduce our wonderful speaker for tonight, Professor Rubén Fernández-Busnadiego, who has been hired in 2019-20, has been hired to our campus to introduce a technology to this campus, which is called cryo-electron microscopy. And this, as he will no doubt show us, it's an excellent, I would say, very, very important technology, highlighted by a Nobel Prize in 2020, I believe, was it, right? 2020? 17 already. Oh my God, how time flies. I'm getting older, losing much of my hair. So that's just how life is. Now, Rubén originally is from Spain, and he did his PhD here in Germany with a group of Vladan Lukic, one of the first people in Germany and in the world overall to use cryo-electron microscopy to look at the brain, to look at synapses. And then he graduated there and went across the Atlantic to the group of Pietro Di Camilli. Pietro is one of the biggest synapse and brain people in the world still alive, so to say. He's one of the very, very fundamental people in this field that made a lot of discoveries. Much of how we understand the brain comes from Pietro. And Rubén had a couple of really seminal fundamental papers there. I mean, your papers on the synaptosomes, on purified synapses from the brain are still cited like there's no tomorrow. And, you know, they made an epoch. I mean, those really were seminal works. And then, instead of staying in the US, Rubén decided to come back to Germany, come back to München, in the Institute of Wolfgang Baumeister there. The pioneer in Germany for this technology, which I mentioned, cryo-EM, and there he had a very successful group leader position for a few years, and then finally joined our campus to strengthen us, to bring this technology and really allow us to see things that there's nothing else out there that can show them to you. So he will show us very small objects, invisible to most other techniques, but coming to life in his hands. So, you know, I don't want to explain how this works, but it's probably the most exciting technology that we have today. So I look forward to the talk. Thanks a lot.
So, yeah, can you hear me? Yes, I think so. Thanks so much, Silvio, for this very nice introduction. Thanks, everyone, for coming here tonight. The lecture is going to be in English. If you have questions in German, I think I can manage. Yeah, today I want to share with you how developments in electron microscopy technology are allowing us to see fundamental or to make fundamental new discoveries into the physiology of our bodies and also understanding disease mechanisms better. Well, Silvio already went through this, but I just want to tell you a little bit more about my story. So I cannot really point very well in this geometry, but I was raised in Spain where I studied physics, and then I went to all these different labs around the world where I was introduced to biological research and also electron microscopy technology. And in 2019, I joined the University Medical School here in Göttingen. And a couple of years ago, in 2021, with the occasion of the inauguration of our lab, we made a little advertising video, and I want to share this with you today because I think it gives you an idea of the machines we are using, of our technology, and the kind of work we do. So I'm afraid the music might be a little bit on the loud side, but let's see how this goes.
The Göttingen Multiscale Bioimaging Excellence Cluster aims to decipher the molecular organization of functional units that are relevant to disease in excitable cells, such as neurons and cardiomyocytes. Cryo-electron microscopy is central to this vision as it bridges the scales between the nano and the micro world, from molecules to cells and even tissues. At the University of Göttingen, we have set up a state-of-the-art cryo-EM platform, which includes equipment for sample preparation, cryo-light correlative microscopy, cryo-focused ion beam milling, and a high-end cryo-transmission electron microscope. Using these instruments, we can image macromolecules at very high resolution, both isolated in their purified form, but also within their native cellular environments. We're grateful to everyone who made this possible, and of course we're excited to start revealing the structural mechanisms that allow excitable cells to work and to investigate the structural causes of their dysfunction in disease.
All right, so that's us, and maybe, you know, looking at this video and from the stereotypical view one has of scientists, one can think this is mainly how scientists look like, you know, with this very sleek, high-tech world, you know, working on very complicated scientific challenges. So there is some truth to that, but there is actually a lot of that as well, you know, just being bombarded with lots of tasks which, you know, sometimes have something to do with science and other times not that much. And actually, you know, when I came here to Göttingen, my first task was to work on a construction site, to put together the facility that you have just seen, and that was not a trivial task because these microscopes are on one hand very expensive, so we had to raise a substantial amount of money to be able to purchase them first. And then also, these microscopes are very sensitive to the environmental conditions, so we had to plan the construction of our facility very carefully. And we started this process in 2019, and everything was going okay. But in 2020, a year later, something got in our way, and maybe you can guess what that was. This was, probably you recognize what this is. This is a SARS-CoV-2 virion, and I'm sure all of you have also stories of how this guy impacted in your life and how the pandemic changed the plans you may have had. So I think this was a pretty traumatic event for the whole world, and I feel now that the worst is over. We're just trying to move on and kind of forget what happened. But in my opinion, our duty as scientists and maybe also as a society is to learn as much as we can from this traumatic event so that we are better prepared for the future. So the topic today is not public health. So I want to focus on lessons that I think are important and that I think we learned or we should have learned from the pandemic. And I want to say that the first of these lessons is that fundamental research is important, and it has a direct impact in our daily lives, often in ways that we cannot anticipate. And this was recognized last year by the Nobel Prize in Medicine to Katalin Karikó and Drew Weissman for research they did some 3 decades ago on RNA modification. So back then, these experiments were seen as kind of obscure research with unclear impact, but it turns out that these RNA modifications that they pioneered enabled the development of mRNA vaccines, which saved millions of lives during the pandemic. So I don't know if you remember how these vaccines work, what is it that they do, but basically, I mean, you see this virion, it has all these spikes. These are actually called the spike proteins. And what the vaccines do is make our cells produce such spikes in isolation. So the spikes alone do not lead to an infection, but they train our immune system to recognize the virus once we're exposed to it. So you may be wondering why I'm telling you all of this, and you think you just walked into the wrong lecture, but you know, just bear with me for a second. I'm telling you this to make the next point I wanted to convey today, that within fundamental research, I think the kind of work we're doing in structural biology has also been quite important during the pandemic. So that what we're doing in structural biology is to understand the structure of proteins and how they are arranged and distributed within their cellular and native environments, And I think also, you know, the research that took place also in the early days of the pandemic has shown the importance of this type of research. So what you see here on the left is a magnified view of one of these spikes. And if you look at the data at the bottom of the reference, you can see all this work was done in 2020. So months and even weeks after the genomic sequence of the virus was available, many labs worldwide were already determining structures that in the end were actually very important to establish the development of vaccines and all kinds of therapeutics. Oops. Right. So if we look at the structure of the spike, one of the first things that scientists found is that it has an important conformational change, and it can go from this closed conformation that you see on the left to an open conformation, and maybe you see that this little domain here is just going a little bit upwards. And this little movement in this protein is actually of very high importance in physiology, because it turns this spike into a key. And this key can now fit into the lock of human cells. And this was also visualized a few months later. This is the lock. This pink part is the ACE2 receptor of human cells that can be opened with this key the virus has. So a few months later in the pandemic, these variants started to emerge that were more infectious than the ancestral strains. And if we look, for example, at the Omicron spike, we can understand why these variants were more infectious. You can see these red balls are the points where mutations have happened, and you can see that in the Omicron spike, the mutations are accumulated in this little piece here, which is a receptor binding domain, which is exactly that part the spike uses as a key to enter our cells. So we can see the key inside the lock today with atomic detail, with atomic precision, and we can understand how these mutations make the key fit even better in the lock, and therefore we can understand why these variants are more infectious than their predecessors. And using structural biology, we not only can see what the virus is doing, but we can also understand how we can start to fight it. So again, also in the very early days of the pandemic, the first structures of the spike protein being bound by neutralizing antibodies were determined, again with a very high level of detail. And not everything was done on the spike, but also on other parts of the virus, for example, the polymerase, which allows the replication of the virus, and several groups, and I want to highlight in particular this work here, which was done in Göttingen by the group of Patrick Kramer, also within MBExC. So several groups were able to determine the structures of this polymerase in complex with antiviral substances. And these structures inform us of how these drugs work, how they impair the replication of the virus, and perhaps what's more important, they inform us on how we can design better drugs that function more efficiently and with lower side effects. So in MBExC, we believe that research, and in particular microscopy, must have a multi-scale dimension. So it is very important to understand biological phenomena at atomic detail, as shown here, but it's also very important to see these proteins in their native context, to understand their distribution and to understand their behavior. So if we want to zoom out a little bit from the single protein and look at their complete arrangement within a virion, then we need to use different techniques, and these technologies allow us to analyze samples that are more pleomorphic. So when we look at the spikes, all of them are more or less the same. But if we look at the virions, we see that they are very heterogeneous. All of them have a different shape, all of them have a different number, a different distribution of spikes. But also these kind of samples we have been, or groups worldwide have been able to image in quite a bit of detail. And actually, if we move further in the scales, we can also see how these viruses actually infect cells and how they replicate inside them. The reason I'm telling you all this is that all these images that I have been showing you so far, from the isolated spike proteins to the virions to these infected cells and the replication of the viruses inside the cells, all this data has been recorded using the same technology, cryo-electron microscopy, different forms of cryo-electron microscopy, and this is the main topic of the lecture today. And I believe this is also, as Silvio mentioned, a very important technology in this mission of MBExC of bridging the scales, of going from the atomic detail to really the molecular, to the molecular details within cells and even within tissues. As I said, this technology is called cryo-electron microscopy or electron cryomicroscopy, and it allows imaging of biological samples in close to native conditions, at high resolution, and in 3 dimensions. So what I want to do today is basically just go across the 3 words that make the name of this technology. So I want to explain to you why we're using electrons, what does cryo mean, and how we do this microscopy. So the first topic, I think it's relatively simple. So why do we use electrons? Because we want to achieve very high resolution. So if we look at this old formula from Abe, we can see that the resolution here, D, is directly proportional to the wavelength of the radiation that we are using in our microscopy. If we're using visible light, the wavelength we are using is of a few hundreds of nanometers. So if we want to increase the resolution, one of the things we can do is just to increase, to reduce the wavelength. So when we are working with electrons, the voltage that we use in our microscopes, you can see that the wavelength of the radiation is several orders of magnitude lower than visible light, and therefore we can reach much higher resolution. There are also, of course, other advantages of electrons and disadvantages, but I think this is perhaps the most important thing to discuss. And in the next slide, I'm going to show you a movie of how actually different techniques come together. And this is going to be, we're going to start with the magnetic resonance imaging of the whole brain. And this is actually the brain of a group member, Felix Boerlein, who also made this movie. So, and now we're going to zoom in progressively. And as we zoom in, we're going to see that at some point the pixels do not contain enough resolution anymore. So that technique was very good to look at the whole organ, but if we want to see, for example, individual neurons, we need to go to a different technology. And this is what we're seeing here using light microscopy. Using light microscopy, we can see individual neurons, and we can also go inside neurons and look at and see, you know, their subcellular organization. But again, we're going to face the same problem. The more we zoom in, at some point, we're going to hit this resolution limit of the technology, and at some point, the pixels are just not going to contain enough information. So we need to change technology again. And now we're already jumping to cryo-electron microscopy, in this case, cryo-electron tomography, where we can see already the organelles. For example, here's a mitochondrion, here's the nuclear envelope, already in a much higher degree of detail. But maybe we want to zoom in further and want to see, we want to study, for example, these macromolecules. Those were individual ribosomes. And for that, we changed technique again. And now we're going to look at single particle cryo-electron microscopy, where we can resolve the structures of protein complexes to close to atomic resolution. So I'm going to tell you in the next slides how, you know, what technology we need to use to obtain such structures. But first, maybe we take a look at the microscopes themselves, and this is roughly to scale, like a maybe simple light microscope compared to one of our current electron microscopes, and at first sight, they look very different. But actually, conceptually, they are not that different. So both of them have a light source here. This one is producing photons, this one is producing electrons. Then they have lenses and that focus the radiation into the specimen. And here's the specimen in the electron microscopy column. Then there are some more lenses, and at the very end, there is a detector. So conceptually, they're actually quite similar. But of course, technologically, they are very different. Sources of photons and electrons are very different. This is not a light bulb, for example. This is what we use to generate electrons in our microscopes, a field emission gun. The lenses are also very different. We don't use glass like in optical microscopes, but magnets that can deflect electrons because they are, of course, charged particles. The detectors are, of course, also different. It's different to detect photons and electrons. But in my opinion, maybe the main technological difference is this property of electrons that they need to propagate in vacuum. So we need a very high vacuum within the electron microscope to allow the propagation of electrons, because if we would have just the electrons going within the air, there would be too much scattering, and we wouldn't have sufficient electrons arriving to our sample and to our detector. So we need to keep everything inside the microscope at very high vacuum, and this includes as well the sample. And this poses a technological challenge that I want to discuss now with the next part of the name of this technology. So we have cryo-electron microscopy. We talked about electrons, how that we use electrons to go to high resolution. Now I'm going to discuss the cryo part, which enables us to look at these cells in close to native conditions. So for that, I want to maybe bring to your attention this movie, which you may or may not have seen. This was like a big thing when I was kind of young, in the 90s or so. And in this movie, Arnold goes to Mars, and somehow he starts to mess around with the wrong people. And he gets into this mountain, and things happen, and somehow he gets expelled from the mountain, from this interior of Mars, and he gets exposed to the surface of Mars. And in Mars, there is an atmosphere, of course. It's not vacuum, but let's think of it as something that is pretty close to vacuum. And as you can see, Arnold is not very happy on the surface of Mars. So Arnold is not very happy in something that is quite similar to vacuum. And that doesn't apply only to Arnold. It applies to actually any biological sample. And the reason is that biological samples, including humans, are mainly made of liquid water. And if we put liquid water inside high vacuum, water will sublimate and samples will be destroyed. So we have this problem that we have samples that are mainly made of liquid water, but we cannot put liquid water inside an electron microscope. So, how can we then analyze such specimens by electron microscopy? So what can we do with the liquid water of our samples? And historically, the first answer to this question was, let's just get rid of the water. So for example, this slide shows the procedure that is used to, or one of the procedures that is most commonly used to get rid of the water of cells. Here we have cells in culture, and we're going to apply a chemical fixative. And what this fixative does is to build a scaffold in the cell that tries to hold things more or less in place. Once the cells are fixed, we will dehydrate them, so we will remove all the water from the cells and substitute this water by a plastic resin. This plastic is then hardened, sliced mechanically using a knife, and this results in sections that can be placed on an electron microscopy grid. Lastly, these sections are contrasted using heavy metal stains. And in this contrasted state, they can be loaded into an electron microscope and analyzed. So even though we are introducing quite a bit of distortions into the sample using this technology. This method of sample preparation has been incredibly useful over the last decades and has shown us most of the things we know actually about the organization of the cells. So this preparation has been incredibly useful in revealing the ultrastructure of the cell, its general organization. But it also has its limitations, of course, and if we want to look at individual macromolecules, we cannot do it this way. So this gentleman here, Jacques Dubochet, who got the Nobel Prize in chemistry a few years ago for developing techniques of doing something else with the water of these samples, he used to say, maybe he still says this, that if you want to study macromolecules within a cell that you have dehydrated, where you have removed the water, this is something similar to studying the distribution of fish in an aquarium after removing the water. So you know what happens to the fish if you do that. So similar things happen to proteins inside a cell when the water is removed. They also kind of fall down and stick to whatever is standing around. So these are meant to be proteins that are happily floating in the cell. Here we have perhaps some cytoskeleton. And when the water is removed, these proteins will tend to stick to each other or to these kind of scaffolds. And actually, when we look at dehydrated cells, this is what we see, but we are not looking at the native organization of these macromolecules. So dehydration leads to aggregation, and this is a problematic artifact. So if we want to understand the native molecular organization of cells, we need to study them in a fully hydrated state. But as we learned, liquid water is not compatible with high vacuum, so we need to make this water solid. We need to freeze it. Now we face the next problem. If we freeze something in a conventional way, let's say we take our cells and we put them in the freezer, this is also problematic. So if we freeze samples slowly, that will lead to the formation of ice crystals, and ice crystals will have several detrimental effects on our samples. So crystals will typically start to form outside of the cell because that's a more diluted environment, and they will start taking up the liquid water. This will have an osmotic effect on the cells that will try to balance the concentration of water in and outside. So they will start pumping out water, and they will start to dehydrate themselves. And therefore, the concentration of all the solutes that we have inside the cell is going to automatically increase. This has detrimental effects and toxic effects to the cell. And when ice crystals get even bigger, if we keep the cells in the freezer for longer, these crystals will start exerting actually mechanical damage on the cell that might be just too much to repair. So we need to avoid the formation of ice crystals, and the way to do this is to freeze the samples faster than the crystals can form. And if we manage to do this, and for that we need to apply cooling rates of around 100,000 degrees per second, we will achieve so-called vitrification. So we will have a form of ice in the cell which is not crystalline, but it's amorphous. It's a glass-like form of ice in which the molecular organization of the solid is very similar to the molecular organization of the liquid. And this allows the preservation of samples in very close to native conditions, and this allows the structural examination at very high resolution. And by doing so, we also avoid these procedures that we were using, that have been used in classical electron microscopy, where samples are chemically fixed, dehydrated, and then stained with heavy metals. So this is a very high cooling rate, and the goal here is to get basically to liquid nitrogen temperature. This is about minus 200 degrees. However, Mr. Dubochet found that actually with a pretty simple device, it's possible to achieve these kind of cooling rates on very thin specimens. So this is the device he came up with. What you see here is these are some tweezers. And the tweezers are holding an EM grid here at the very bottom. And these tweezers are mounted on a piston that will shoot the specimen into this reservoir that contains typically liquid ethane that has been cooled down by liquid nitrogen at about, as I say, minus 200 degrees. If we put our sample here, for example, some viruses, this solution of viruses or other proteins can be vitrified, can achieve this state of solid amorphous water, and immediately this was recognized as a very important finding with quite high impact in the scientific community. So vitrification is key to all forms of cryo-electron microscopy, but there are some people that like vitrification too much and take it too far. And some people claim that you can actually vitrify yourself when you're sick, and then just stay in one of these tanks for 200 years, and at some point technology will be good enough to cure your incurable disease and bring you back to life and let you live another 100 years. So don't spend your money on this, because as I said, for vitrification we need to achieve this 100,000 degrees per second, and this we can achieve with a simple device on very, very thin specimens, on these suspensions of macromolecules, but we cannot do so on thicker samples. So it's already difficult to vitrify single cells. It's much more difficult to vitrify tissues, and it's essentially impossible, at least with current technology, to vitrify organs and much more whole human body. So, save your money for better technologies. So that was the cryo part of cryo-electron microscopy, and now I want to tell you a little bit about the microscopy itself and how we managed to image these specimens in 3 dimensions using microscopes that actually can only produce flat 2-dimensional images. And so there are 2 main types of specimens that we study in cryo-EM. One are purified proteins, so these proteins can be expressed and purified, and we can have millions of copies of the same macromolecule. And to study these kind of specimens, we use the so-called single particle analysis. And there are other specimens of which we cannot have 2 of the same. So as we saw before, there are not 2 variants that are identical to each other, and there are of course not 2 cells that are identical to each other. And to study that kind of specimens, we use a different form of cryo-electron microscopy that is known as cryo-electron tomography. In single particle analysis, so when we work with purified proteins, this is basically what we have to do. So this schematic is showing the same thing as in the video. We have a solution with our purified proteins. We will apply it on an EM grid, which is held by these tweezers, similar to the Dubochet device. Now we blot the sample, we remove the excess liquid, and we shoot it very quickly into this cryogen. And by doing so, we achieve this very thin layer of, that you can see here in the bottom. We have this very thin layer of amorphous ice, and our proteins of interest are suspended within the layer. And in principle, because the proteins were freely floating in solution, they will be in this layer adopting random orientations. So we will have a slug of ice that you can see here with millions of copies of the same macromolecule in different orientations, and then we will take pictures of it from here. These are our electrons that are imaging this specimen, and because each of them is in a different orientation, we will get different views of very similar objects. Then computationally, we can try to figure out where do these views correspond to in a 3-dimensional structure, and we can reconstruct this structure with a lot of detail. And to illustrate this with a practical example, I want to tell you a little bit about the proteasome. And this is the work of the group of Eri Sakata, also at the University Medical Center Göttingen. The proteasome is the main machine in the cell for targeted degradation of proteins. It's highly conserved, and it basically consists of 2 modules. This regulatory particle here is recognizing substrates, is recognizing what needs to be degraded. So this is kind of this recycling station, just sorting out the trash, and handing over the trash to the next module of the machine. There is this ATPase that uses ATP energy to unfold proteins and feed the proteins into the last unit of the machine, which is this proteolytic core that is basically chopping the proteins into little pieces so that they can then be recycled. And Ari's work, or Ari is very interested in understanding how this machine is built, because only then we can really understand how it works, and only then we can understand how it doesn't work in certain diseases. So her group revealed one of the first high-resolution structures of the proteasome, which you can see here already some years ago. You can see that resolution is local, and that the parts that are more solid, more stable of the protein can achieve higher resolution than the more flexible parts. But another important characteristic of cryo-EM is that when we freeze these machines, they are actually carrying out their work. So they are performing their native function, and like many other machines, like a car or anything else, they need to move to carry out this function. When we freeze them, we have snapshots of individual particles in different stages of this conformational cycle that they need to undergo to carry out this function. And Ari's work has also revealed the most important stages of this conformational landscape of the proteasome. So the proteasome has to undergo these major conformational changes to recognize the substrates, unfold them, and then chop them into little pieces. So this is basically what single particle analysis does. We can obtain a very detailed blueprint of these machines. We can understand how they move, but we do not see how they work in their native environment within cells. And to do that, we resort to cryo-electron tomography. When we have specimens that are unique, we don't have millions of copies of the same cell because each cell is unique. We need to image them in a different way. And for that, so this is our specimen, and what we will do is we take pictures of it from different directions by tilting the specimen within the microscope, and then we end up with this series of pictures that we can reconstruct into a three-dimensional object. And now, moving to the topic that we are working on in my laboratory, we mainly use cryo-electron tomography to study some of the effects of aging. So this was Arnold in the 90s, and this is him a few decades later. And of course, aging has very profound effects on our organisms, and now we understand that it is actually the major risk factor for many diseases. And this includes neurodegenerative diseases, which, as you know, affect mainly our last decades of life. And this is a big problem, especially in our societies, where the average age of the population is increasing. So it is estimated that nowadays around 7 million people in Europe suffer from Alzheimer's or related dementias. Alzheimer's is the most prevalent of these diseases, and this prevalence of the disease is, of course, expected to increase as the population ages. And this is a big drama for the 7 million people and their families, and also poses a huge burden a huge financial burden on our healthcare systems. One important aspect of Alzheimer's and other neurodegenerative diseases like Parkinson's and others is protein aggregation. So we just saw before how proteins need to achieve their native structure that we can study with cryo-EM, and this native structure is necessary so that they can carry out their function. In many of these diseases, proteins fail to achieve this native structure. They misfold, and when they misfold, they aggregate, and they form these mesoscopic structures that pathologists can observe in the brain of patients that died of these diseases. And the driver of aggregation in each of these diseases is a different protein, but somehow they all end up going down the same cascade. And what I think is amazing is that we know this for more than 100 years. So these are original drawings from Alois Alzheimer more than 100 years ago. So we know that aggregation is a main characteristic of Alzheimer's and these diseases, but we still do not understand the role that protein aggregation plays in disease. So we do not know to what extent these aggregates are the causal agent of the disease, and we do not understand whether these are really the main therapeutic target that we should be addressing. And this is one of the questions that we want to investigate in my laboratory. And to do that, we want to image these aggregates within their native cellular environment, within the neurons in which they form, and within the neurons they may eventually contribute to kill. And we want to understand the native structure of these aggregates, and maybe more importantly, how these aggregates interact with their cellular environment. Because we think that if we understand these interactions, we will understand better the contribution of these aggregates to disease. And to do so, we use cryo-electron tomography. And how we do that is shown in this movie. So here we have a living human cell that contains one of these aggregates that we mark fluorescently. This is a polyglutamine aggregate found, for example, in Huntington's disease and others. We have a living cell, and now we're going to freeze it. We're going to vitrify it at liquid nitrogen temperature. And from this point on, we're going to keep it at this temperature at all times. The first thing we need to do is we need to thin down the cell, specifically at the location of this aggregate. And for that, we are going to use a machine like this one. This is a cryo-focused ion beam scanning electron microscope. What you're seeing here is the loading of the samples in this shuttle that contains 2 EM grids. And on these grids, we have vitrified cells containing aggregates. We can ignore this step. It's kind of a technical thing. But now we're going to start using the electron beam of this microscope to perform correlative microscopy. So we want to find back the cell we identified first in the light microscope that contained that green aggregate. We want to find it on this EM grid after vitrification. And it turns out to be this one, which has been frozen on the substrate of the EM grid. And we know from the light microscopy that our aggregate is somewhere there. And now we're going to start using the ion beam to remove material from the cell. Basically, everything that's above and everything that's below this aggregate is going to be removed by the ion beam. And the only thing that remains from the cell is this very thin slice, which is about 100 nanometers thin. And as you can see, it remains suspended by the parts of the cell that have not been touched by the ion beam. In this machine, we only do this step of sample preparation, and then we transfer the samples to the next microscope, to the transmission electron microscope, where we do the high-resolution imaging. And this is shown here. All these transfers between microscopes and everything else, all the imaging is done at liquid nitrogen temperature. To get these three-dimensional images, we take pictures of the specimen from different directions by tilting the stage of the sample. And then we can computationally back project these images to reconstruct the three-dimensional volume of the aggregate. Here, I'm going to show you one example of one of these tomograms. This is this polyglutamine aggregate within a human cell. And we're going to see the raw data. Here we're going up and down in optical slices. And now we're going to see the computational analysis of this data where we can separate the different elements. We can see these blue fibrils of the aggregate and different cellular components in other colors. Today I want to tell you a little bit more about a disease that is called amyotrophic lateral sclerosis and a related condition known as frontotemporal dementia. This is the third most common neurodegenerative disease and affects mainly motor neurons. So this is not always linked to dementia, to cognitive problems, but it's linked to impaired muscle function. And it's a really devastating disease because life expectancy is only a few years typically after diagnosis, although there are cases in which patients manage to survive for much longer, but this is really exceptional. Most ALS patients also suffer protein aggregation in their brains. Most cases are sporadic, and in those cases there is aggregation of a protein called TDP43. But there are also familial cases, and most of those patients suffer a mutation in this gene called C9orf72. And the main driver of aggregation here are proteins called poly-GA, so these are just repeats of glycine-alanine residues up to hundreds or thousands of times. So I want to show you some work we've been doing with this kind of aggregates within neurons. We can use this workflow I mentioned before to take these kind of tomograms. So now again, we're going up and down in this volume of the tomogram. We can see the aggregate, and now you'll see the result of a computational analysis separating the main macromolecules found in this volume. So you see the aggregate in red. It forms kind of planar, twisted ribbons. You see also some other macromolecules like ribosomes here in yellow. And here in purple is the trig chaperonin. And maybe you could see that those macromolecules are mainly found in the periphery of the aggregate. But what was really dramatic in this case was the accumulation of these green macromolecules within the aggregate volume. And directly from their structure, we can tell that these green macromolecules are proteasomes. And we can do so because the sample has been preserved in these close to native conditions where we can recognize macromolecules directly from their structure. So we found that within these aggregates, there was a concentration of proteasomes that was 30 times higher than in control neurons. And this was very interesting because proteasome dysfunction has been implicated in many diseases, including many neurodegenerative diseases. So I showed you before how this single particle cryo-EM work from Aries group revealed this conformational dynamics of the proteasome. And this work was done in vitro with purified proteins, but now we can try to find all these different conformations also within the neurons. So what we can do with this type of data is we have here these individual proteasomes, and we can extract them from their cellular environment, and we can align them and average them like we do with purified proteins. And then we can also try to separate all these different conformational states. The result of those such analysis in these aggregates is shown here. We have these 4 main classes of proteins, and we could assign their functional or conformational states. We could see that these green groups of proteins corresponded to the ground state of the proteasome, where it's not engaging in substrate degradation. This other group of proteasomes corresponded to the so-called S2 state. And finally, this group corresponded to the so-called S4 state. And this is what we found most interesting. Because in vitro, Aries' work showed that this S4 state is a very transient conformation, and we can only observe this conformation if the proteasome is stuck with nucleotide analogs that cannot be hydrolyzed efficiently. However, in our samples, in these aggregates within the neurons, we found that almost a quarter of the particles were adopting this conformation that is supposed to be very transient, suggesting that perhaps the proteasome is kind of getting stuck at this particular time of its conformational cycle. And what we can do with these tomograms, what I told you before, is we find individual particles, we extract them, we figure out their conformation. We know now the conformational, the functional state of each individual particle, and now we can put them back. We can put them back into their cellular volume, and we can see if these different states follow any particular patterns. One of the things we measured was the distance between each proteasome and the closest aggregate. And this is shown here in this graph. You see 4 dotted lines, and these are these 4 conformations that I showed you before. And this cyan line is this S4 class. And if we look at proteasomes that are within 15 nanometers from an aggregate, like these ones here that are essentially touching the aggregate, we can see that about 40% of them are in this particular conformation. And if we move away from the aggregate by only 30 nanometers, so this is less than the length of one individual proteasome, we can already see that the fraction of particles in this conformation has been reduced to half, indicating that it is really this direct interaction between the proteasome and the aggregate which is stabilizing this particular conformation, indicating again that the proteasome is getting stuck because of this interaction with the aggregate, which we later proved using functional assays that I will not describe here. But I think what was important is that also our collaborators could corroborate these findings in patient tissue. So here you can see that a patient that died suffering this mutation, if we look at the proteasomes within this brain, most of them colocalize actually with this aggregate. So this is a phenomenon that is actually happening in the brains of these patients, and this may hopefully have some therapeutic relevance as well, because preclinical experiments have shown that if we try to counteract the effect of these aggregates, if we pharmacologically activate the proteasome, we see an improvement in cells or mice that carry these aggregates. So I mentioned that there are familial and sporadic forms of the disease. I told you about the familial cases, and now very briefly about some work we have done on this TDP43 aggregation that is characteristic of sporadic cases. This is one tomogram recorded in a neuron that has one of these TDP43 aggregates. Now again, we were going to see the macromolecules. In red is the aggregate. The aggregate itself is very different from poly-GA. Now it doesn't form these ribbons anymore. It's more of an amorphous structure. But we see that there is also a profound accumulation of proteasomes that are shown here in blue within the aggregate volume. And again, we could show that these proteasomes were impaired, were functionally stuck by this interaction with the aggregate. So we could show that both for aggregates that are characteristic of familial cases, but also for sporadic cases of ALS and frontotemporal dementia, we have different proteins that are aggregating. These aggregates have a different structure, but somehow they mess around with the neuron in a similar way. They attract proteasomes, they recruit them, and they get them stuck in there. The effect is that this fundamental machinery of the cell that is taking care of the trash, of recycling, is not correctly functioning. And what we have in the end in these neurons is an accumulation of trash, which of course has many detrimental effects and can contribute to toxicity and to the neurodegeneration that we observe in these diseases. So with this, I think I would like to finish. These are the take-home messages that hopefully I could explain to you that vitrification, this very fast freezing, preserves biological samples in close to native conditions. If we are working with repetitive structures, such as purified proteins, we can image them by single particle cryo-EM at atomic or close to atomic resolution, and I told you a little bit about the proteasome work in that context. If we are looking with more complex, we are working with more complex specimens such as cells, we can image them by cryo-electron tomography. And with this, so I illustrated this with this example of the protein aggregates related to amyotrophic lateral sclerosis and frontotemporal dementia, how they recruit and store proteasomes. So hopefully I can show you how we're trying to do what we call structural cell biology using these techniques. So how we can start to understand basic fundamental cellular phenomena and also how these phenomena are altered in pathological conditions directly from a structural perspective, directly in situ within cells. And globally, I also hope I could also convince you how cryo-electron microscopy is emerging as a powerful tool in biomedical research, addressing problems from viral infection to neurodegeneration. With this, I'd like to finish thanking my group. Here's a picture of my group and Ares together. Most of the work I've shown here was done by Chang when he was a postdoc in the lab. Now he has his own lab in China. These are our funding sources, our collaborators, to which we are also very thankful. And of course, I'm very thankful to you for your attention. Thank you very much.
And I'm sure we are all eager to ask questions. So, yes, we have a question. That's okay. Okay, you want to do the running around? Okay, that's perfect. Then I have
this is super fascinating. I'm working for Hoffmann La Roche in Basel and have some insights into some of the diseases in this area. I'm just wondering, when you see at the sheer number of aggregated proteins in there, when you think about proteasomal activation principles, right? You know, at a certain point in this aggregation accumulation, there is a point of no return, right? Likely where even proteasomal activation may not really change course of disease anymore. Have you got any ideas about, the quantification and how much of, this aggregation will then, in a certain stage of a disease, where still an intervention may be feasible?
Yeah, so there's probably a point of no return in terms of aggregation, but we don't know if there is a point of no return in terms of proteasomal stalling. So our idea is that activating the proteasome may or may not help aggregation per se, but it will help the cell to keep its recycling machinery active. So other substrates beyond this aggregating protein, other substrates that need to be recycled will be able to be recycled within the cell. So this wouldn't be a therapy that will cure the disease. I think this is not something that anyone is thinking of for the moment for any of these neurodegenerative diseases. The hope is that one can prolong basic quality of life by helping the neurons cope better with these misfolded aggregates. Yeah, I mean, prolongation, let's say, yeah.
Other questions? I actually have one, and this is really a sneaky question. Did you get some help from Alexei Chizik on these wonderful movies that you've shown, or who made them?
Yeah, no, I mean, these movies are coming from different sources. Okay. But the one where, for example, we explained the workflow of tomography, this we did together with Thermo Fisher. Okay. So they were making, a very nice promo movie for their microscopes, and we just sneaked in our aggregates in there, and could collaborate with them.
Wonderful.
Nicely.
No, because we actually have a specialist in Göttingen that did some movies for some other people, very, very similar to your Thermo Fisher movie. Astoundingly similar. So that's why I asked. Yeah.
Okay.
Thanks a lot. Okay. Other points? Yes.
I wanted to ask you about if you have studied different stages of those aggregates in the cells, and if so, I'm curious about where do you get those samples from? Are they human samples?
So different stages we have not directly addressed because it's not easy to temporarily control the aggregation of proteins. We have looked kind of at early stages of aggregation and late stages of aggregation, and we have not seen qualitative differences. So of course aggregates get bigger. The bigger they are, the more, for example, proteasomes they sequester, but we can see that phenomenon already with the smaller ones. The samples I have shown today are not human. So these are cultured primary neurons that are expressing aggregating proteins. But actually now one of the main drivers of our activity is bringing this to human systems that might be more directly relevant to what might be happening in the brains of patients. And for example, we're doing a lot of this work now with alpha-synuclein aggregates, which are found in Parkinson's disease. In collaboration with our colleagues in the clinic, we can obtain these aggregates directly from patients, either from patients that passed away already with the disease from their brains, but also from living patients, because we can obtain aggregates actually from the cerebrospinal fluid of living patients. And the hope there is that by examining aggregates of patients at different stages of disease, maybe even within one patient during disease progression, maybe we can also understand with our system how this disease is progressing and how the aggregates are maybe interacting with the cellular environment in different ways. So this is definitely something that we are pushing for the present and future work. Yeah.
Thank you.
Thank you.
I mean, that sounds great, but in CSF, you have a lot of different things. I mean, so how do you recognize that those are synuclein particles?
I mean, what we are using the CSF for is to seed aggregation in neurons.
Seed aggregation.
Yeah. So we look at intracellular aggregates that have been triggered by exogenous aggregates obtained from patients.
This is a great technique. I mean, there's a couple of diagnoses for Parkinson that have been made with this last year FDA approved in the US. So yeah, very cool, very cool.
Have there been studies on mice or other animal models where you can use gene therapy or something to introduce to rescue kind of a good copy of functional proteasome?
Right, so I mean this, for example, this pharmacological activation of the proteasome, this has been tried by our collaborator in Munich using mouse models that, express this aggregating protein, and just by feeding the mice a drug that is known to activate the proteasome, the mice basically get better. So these are kind of first steps into therapeutic workflows, but this is kind of ongoing. Yeah.
But not yet gene therapy or something that creates a...
No, I mean, I think gene therapy is probably something one would like to avoid if there are simpler alternatives. So if one can use a pharmacological approach, this I think is often the first thing to try. Yeah, so this is, I wouldn't say this is a direct candidate for gene therapy, or I wouldn't know now, how to use it. But yeah, definitely in other diseases, this might be a very important approach. Yeah.
Okay, then I think some of our audience members indicated it's time to wrap up. So yeah, thank you very much again, Rubén. So please
thank you very much.
Give him a hand. And yes, last words, because we are a little bit late, wir sind schon ein bisschen spät, aber nächste Woche, 23. Januar, werden wir unser nächstes Termin haben, unsere nächste Vorlesung, dieses Mal von Professor Niels Voigt, auch Universitätsmedizin Göttingen. Er ist ein Herzspezialist und jetzt werden wir über den Herz sprechen. Herzbeben, wenn das Herz außer Takt gerät. Wahrscheinlich pathologisch, nicht nur physiologisch und sagen wir, nicht nur wegen Liebe oder sowas, wahrscheinlich auch wegen bestimmter Krankheiten. Vielen Dank und bis nächste Woche.